Okay, great. Welcome everyone to April's Reclone community meeting. Today we are joined by our fellows from the Reclone regional hubs. So from our Argentinian hub, we have Romi Russi, who's an assistant research fellow postdoc at the National University of Cuyo, sorry. And also Diego del Balzo, who is a doctoral fellow also at Cuyo. In Ghana, from our Ghanaian hub, we have Prince, who is a research lab manager at Ashesi University. And then in the Philippines, we have Aaron Macauyag, who is the lead scientist at the Integrated Protein Research Development Center, and co-PI at the Ateneo, the Manila University, and also Relles, who is also a research fellow at Ateneo. So without much further ado, I'm just going to pass on to the fellows to tell more about what they've been doing at each of their regional hubs. And I think to start off, I think we have Prince, who will be telling us the work that they've been doing. So Prince, take it away.
All right, thank you very much. I'm going to share my slides. That's okay. I just one bit. All right, thank you again. I'm happy to be here. My name is Prince Samoh. I'm one of the fellows, and I'm the fellow for the Ghana Hub. So a little background about my experience. I have a background in biomedical engineering from University of Ghana. And then from there, I worked as a co-founder and then a research lead on a community lab that we created in Ghana called the High Bio Lab, which over time came to work with the OBL, Open Bioeconomy Lab, which is being headed by Jenny Molloy. And I worked in this lab from August 2020 to July 2022. Sorry. Right. And then from there, I moved to join the High Bio Lab, Ashesi University Bioengineering Lab with Elena, where I manage the bioengineering lab. I assist with the synthetic biology class, as well as advice on most of the project that goes on in the lab, including the iGEM project. And then talking about iGEM, I've been a part of iGEM since 2021, first as a team member, and then ambassador in charge, and then judge and advisor to a lot of teams last year and this year. And then, so my first role with Reclone was in 2021, where I was asked to give a presentation on democratizing the means of biotechnology for global access. So this was our iGEM project in 2021, where we were trying to look at how do we make biotechnology accessible to a lot of people, especially in the global south. And so that was my first presentation as a part of the Reclone. And then when I moved from the High Bio Lab to Ashesi University, that's when the Reclone then also moved with me to Ashesi University, where we are currently running one of the hubs here. Then I became a fellow in 2023, where mostly what we've been trying to do is kind of introduce people to what Reclone is. Elena has done a couple of presentations to people about what the Reclone is and trying to get people to understand how to use the parts and what these parts are meant for. But one big challenge that we kind of looked at in this part of the region is that there is a lack of skills on how to actually even use this parts in the first place, right? Like you can talk to people about what the parts are, what you can use them for, but then the skills to actually, work with it and the resources to work with it has quite been a little bit of a challenge. So aside that, we have tried working on a couple of things to contribute to the Reclone collection, the OBL collection, where we started a project which intends to basically design a DNA ladder. So it's just going to be like a plasmid where we send the plasmid to people and then it just digests the plasmid and you get a DNA ladder. So what we did was we basically just used the parts in the open DNA collection where we just PCR some part from it. And then we assemble it using Golden Gates to then, once we have that assembled, we digest with BSA1 to get the ladder profile. So this is one of the activities that we are currently doing to kind of contribute a bit to the collection that Reclone has. Instead of just enzyme, we can also include DNA ladder as part of some of the things that OBO does. So on that front, we've done quite a lot of things. So as I said, we didn't synthesize the parts as a whole. We just pieced out most of the parts from the OBO collection. We assembled. We did PCR to confirm if the assembly was successful, and then we did digestions to see what it was actually like. But we started having a bit of a challenge because we weren't getting the profile right. The profile we were looking for, we weren't getting that right profile, and so we just did nanopore sequencing and then looked at an alignment. We did an alignment with the original sequence on BenchLink. We realized that everything was right except some particular regions where there were some mutations and then some deletions. However, most of the mutations happened in a region where the BSA1 wasn't present there. So we weren't worried about most of the mutations that occurred, most of the mutations and deletions that occurred in the assembled part. But one critical one was a portion here where we have the BSA1, which is supposed to cut. So we realized that this portion here had some mutations and some deletions, and so that could contribute to why our digestion wasn't working. But even with that, we're expecting that the profile will still be there, but not exactly the number that we're looking for. And so we tried several times and we weren't getting that profile. And so what we realized was, or what some of the troubleshooting we kind of looked at was that probably could be due to the fact that the plasmid was super folded because it was 16,600 base pairs, which is pretty big. And then also we kind of looked at the fact that it could be methylated. And so we had to troubleshoot by changing the strain. So we've actually gotten non-methylated strains that we're actually using to kind of troubleshoot to see if indeed it was due to the fact that these sites have been methylated, and that's why the restriction enzyme wasn't working. So once we checked that out, we'll be able to figure out whether it was methylation that was making the enzyme not cut, or it was the superfolding, and then we can figure out what to do from there. Aside that, we've worked on a couple of hardware projects as part of contributing to the open science. We've built open source incubator, calorimeter. So the open source incubator was what we were using in the high bio lab because we didn't have the funds to buy a commercial incubator. So that was what we're using to do most of our experiments. And then we had calorimeter. So we built this because at first we're using the McFarland standard to kind of measure what our ODs were. And then we figured that, if we were able to build this, then it's going to help us, kind of close the gap between, how sensitive our experiments are. So we did that and it works all right. And then we also worked with open flexion microscope to build a couple of microscopes as well. So, kind of just brushing on what other things that we've been doing aside, Reclone, what other open science activities we've been doing. So we last year gave a presentation at the Open Science Festival in Germany about what it is like doing open science in this part of the world. And then also we gave a Reclone presentation on how do we use the open enzyme collection and what are the benefits of it and how best do you use that to for frugal biomanufacturing. And then we've been a part of the Global Community Bio Summits, giving presentations on how we do open science research in Africa. And then we've had a couple of enzyme manufacturing training because we realized that we can give people these parts all right, but if you don't kind of help them figure out how to utilize it, how to produce it, it gets very problematic because then it just sits in the lab and then they don't do anything about it. So these are some of the things that we came up with to see how best we can actually figure out how to help the people in our region. So what we've been doing since for the past year was we received the parts. We've had some interest in the parts. What we tried to do was to express the enzyme because, again, as I said, we have to train people on how to express the enzyme, and we plan to have trainings where we are going to not just give you the part, but also help you figure out how do you express the proteins, how do you check for the quality and all that. And another thing is that one of our students actually got a grant, the health initiative grant, where we are looking at producing this enzyme. So if somebody doesn't have the facility to produce these enzymes, what we want to do with this grant is to basically produce these enzymes, purify them, whether as purified or as the cellular reagents, and actually hand them out to people to actually use for their experiment. And so these are some of the things that we've been have been able to do, and some of the things that we are currently looking at doing in order to really assist people with the Reclone collection. And then finally, all this work at the hub here has not been possible if not for Elena, who has been an instrumental part of the Reclone community, and also helping us with everything we need to make our hub work. And so thank you to Elena. Thank you very much.
Great, thank you, Prince, for that rundown of the Ghana Hub. It seems like you do a lot more beyond what you do at Reclone, definitely. And we're going to leave the question, or feel free to put your questions in chat, but we're going to go through the other two hubs first, the fellows from there, and then we'll leave the end of the meeting for any questions. So next up, I think we have Aaron and Moy talking about how the Philippine hub.
Okay, so I hope everyone can see my slide. Yeah, okay. So hi, good day, good evening from the Philippines. So we are from the Reclone Hub from the Philippines in Asia. Okay, so as you can see in the map, the Philippines is actually an archipelago, which makes it a lot harder to transport and bring out reagents from the capital city, which is in Manila. And the Reclone Hub is currently housed in Ateneo de Manila University, that's in Quezon City, in Metro Manila, which is in the Luzon Island in the Philippines. And currently, most of biotech research are only centered in the capital of the Philippines, and the rest of the island actually lags behind biotech research, especially on medical diagnostics. So as an introduction, the Reclone Hub in the Philippines is currently housed in a program in Ateneo de Manila, which is called the Integrated Protein Research and Development Center, which is a biotechnology facility for health. The IPRDC, as we call it, is a national center of excellence in applied protein research and development in manufacturing in the Philippines, which has a goal of providing reagents for all. So the center actually has 4 parts in it. So the first has 4 teams in it. The first team is the one which we are part of, which is responsible for creating protein out of the plasmids that we get from Reclone. And the second team is on fermentation and fermentation optimization. We also have other teams which work on open source hardware and purification materials. So just as a short intro for the other teams, so you can see what the program or the center is currently doing. We have the team 2, which is currently working on multi-parameter optimization of 5-liter and 10-liter bioreactors, mostly composed of chemical engineers. We also have the third team, which is currently working on bioreactor design and testing from open, what do you call this? Wherein they are on instrumentation creating these bioreactors, which we will also eventually use for the center. And the last team is currently working on magnetic nanoparticle development, which is our materials team. And us in the biochemistry team, we are the ones currently doing the expression of the plasmids that we get from Reclone, and we also get some from Agene, up to extraction, purification, and functional characterization. So our current focus right now is on diagnostic enzymes. Specifically, we are working on polymerases, RTs, and we are also exploring the possibility of working on industrial enzymes and hopefully be able to transfer this technology to our local industry partners. So we've, I think, mentioned quite a few of our partners in our previous community meetings, and these are the people in our team. We have Doc Aaron here with us, and I think also Victor is present in this meeting as well, and these are the rest of our members. So, what are our activities as a Reclone Hub in the Philippines? We were able to do a soft launch of the Reclone Hub last year, November, I think, in the 50th annual convention of the Philippine Society of Biochemistry and Molecular Biology. So we were able to present a poster introducing the Philippine Reclone Hub, and we were able to create a network with other institutes in the Philippines, like the National Institute of MDB. We were also able to create a network with the Philippine Genome Center, the different labs in there, and also other state universities in the country, as well as health institutes who are interested in working with the plasmids that we have from Reclone. And we were able to do a hard launch of the Reclone Hub Philippines in one of Doc Aaron's talk at the Institute of Biology at the University of the Philippines. This is the national university of the country. Last December, and we were able to bring with them the ones that were prepared in the lab there. And we also gave out samples from our collection. So just as an update, because we weren't able to actually prepare a slide for our lab works, we were able to successfully transform all of these plasmids into E. coli BL21. And currently, because we don't have that much manpower, we are only currently able to express and purify the M. Sherry open vent that was part of this collection. So this is what we got here. Here's our pitch poster for the rest of the institutes who would like to avail of the plasmids that we currently have. So we were able to work on M. Cherry Open Vent. We were able to purify it, and tomorrow, this week, or next week, we are going to confirm whether it's working or its functionality is working or not. So we were able to participate in the Policy Pre-Fellowship that Reclone had last year, and we were able to collaborate with these institutes in here. And the data we got was actually helpful, and we were able to present it in one of the committees of biosafety in the Philippines. They had this program where Doc Aaron was able to present our findings in our policy brief, and they were able to point out that the Philippines should push for the prioritization of biotech research, especially in medical diagnostics in the country, because that is currently lacking in the biotech landscape of the country right now. We also pushed for the membership of the Philippines in ICGEB. So I think that was the last slide. So thank you for listening.
Okay, thanks very much, Moy, for that rundown of what you guys have been doing over there. And we'll just wrap it up with our final hub. So this is our Argentinian hub with Romi and Diego. So I'll hand this off to you.
Great. Here we are. Okay. Okay. You can see the screen and you can hear me? Is everything okay?
Yes,
Great.
it's okay.
Okay. Well, my name is, I'm Diego del Balzo. I am a PhD student and I work like a teaching assistant in the Faculty of Medical Science in the National University of Cuyo here in Mendoza. My partner, Romina, is an assistant researcher and is teaching assistant too in the same course than me here in the university. The university is in Mendoza. We are in the west of Argentina, very close to Chile. Our director is Dr. Teresa Damiani, who a few years ago started to be in contact with Dr. Federici, and Dr. Federici introduced Jenny Malloy to Dr. Damiani. This interaction encouraged Dr. Damiani to the idea to have a Reclone hub here in Argentina, in particular in Mendoza, in our university. In this context, in November of last year, we received a diagnostic collection of reclone plasmids in this format, in paper. Our first perspective was obviously generate glycerol stock, obtain the plasmid, and after that, with the plasmid we obtain, we generate new glycerol stock with starting for this second generation of plasmid. And after that, we check concentration, integrity, and at this point, we are in condition to share every plasmid of this collection. And obviously, we have the intention to continue receiving more plasmid and more collection, with the intention to complete the different collection that we can have here in our hub. In terms of expression or purification of this plasmid in particular, we are not working on that at this moment, but we work in expression of other plasmid of Reclone. Romi, if you want to tell about it.
Well, for the moment, we express PFU in two opportunities. The first expression and purification, we start a 20 ml culture, and we purification this enzyme by bit magnetics. And the second induction, we start a 50 ml culture, and the purification was with IMAC resin. And in both cases, the other slide, please. In both cases, we obtain PFU with activity. In the first case, we use PFU twice 5 and 10 dilution. In the second case, we use smaller dilution PFU without activity. So we use currently PFU 10 or 20 time dilution. So thank you, Reclone, because this enzyme is the key reaction for the laboratory, because in our laboratory, doing molecular biology, cellular biology, and the other technique with using this enzyme.
Great. As I mentioned before, our director, Dr. Damiani, have a very strong intention, and we all believe in start to work strongly with open science here in our institution. Fortunately, Dr. Damiani is currently the main responsible of science and international relationships here in the university. In relation with her conviction in open science, last year we organized the first open science symposium here in our university. Every person, every researcher who has one project related to open science in this opportunity could show their work to the students of the university, to other researchers, and to the community in general. In our case, we showed our work trying to be a Reclone hub here in Argentina. And obviously the experience was very important because different persons, different researchers who work in open science spend time together here in the university for the first time. So it was great to generate interaction between all the people who is interested in open science here in the university. And it was very important to show to the community what are we doing in this context. It was a very significant experience, and the idea is to continue doing this symposium, this meeting every year, and with more researchers, with more people who is working in open science here in our project, in our province, in our country, in the best of the cases, in our region in general. My perspective in relation to open science and in particular to Reclone here in our university is continuous working and in the best of the case obtain economic support to sustain the open science here in the university. In an ideal point, we aspire to arrive at the moment in which open science and Reclone in particular are being structural politic here in the university, and at the point it could be independent of Dr. Damiani, Romi, or me, and that is our goal at this point, to continue working in order to open science and Reclone be something stable here in our institution. Thank you very much.
Great, thank you, Diego and Romi, for giving that introduction as well about the Argentinian hub. I'm just going to pass to the audience. Do you have any questions that you want to ask the hubs about? Oops, straight away. Adrian, do you want to ask the question, or do you want me to read it out?
Oh yeah, we can ask that. So I was wondering, Romina, you use the two methods for purification for PFU, the magnetic beads and the resin. Which one worked better in your opinion? And if you have the protocols published or accessible somewhere?
Yes, the IMAC protocol is the conventional protocol we use in the laboratory. In this case, use imidazole
Yes.
as a elution buffer, and the enzyme have activity with the dilution more smaller concentrate, because I think the imidazole can be inhibit the activity. and the expression was in the same condition of induction in terms of temperature and time, but the concentration is bigger. Is good. I can share the protocol in the
That
forum.
forum. Okay, beautiful. Thank you very much. That's exceptional work there. Thank you so much.
No problem. Thank you.
So, I mean, Prince, you started off by saying, well, I think all of you guys touched on educational outreach and kind of open science activities, because you highlighted that as not only providing people the reagents, but also needing to educate and kind of provide them how to do so. What do you think works best when talking to other people about it? Do you think like live demonstrations, practicals? Do you have, well, to all of you guys, do you have the protocols that you can share on the forum as Romi has suggested for her? So basically, what are good outreach techniques?
I could go first. So I would say that I think in our part of the region in Ghana, I think the majority of the problem is on the fact that most of the people that do molecular biology research already get their enzymes from commercial companies outside Ghana, and most of them don't. Because of that, they don't have the resources to actually, produce the enzyme, or even, in terms of the technicalities on how do you produce the enzymes and how do you purify the enzymes, because, they don't have the resources. Most of them don't have incubators, they don't have, most of these things. So the problem then is, if they take this collection, then how do they use it? How is it beneficial to them in the first place, right? And so we realized that, in order to really fully give them what it takes to be able to do that, that's why we even had most of the hardware projects in there as well, because these things are kind of crucial to help you produce the enzyme, even if it's as a cellular reagent that you can just directly put into your reaction to run your PCR or run whatever you need, right? And we had a student work on building a bioreactor using kind of low resources, and that kind of works as well. So we kind of looked at the fact that to be able to really allow them, or give them the full benefit of what the Reclone capabilities of the Reclone parts, we need to also incorporate the hardware components of even if you don't have the funds to buy this expensive equipment, you can do this open source build up as well as looking at how do you give them the skills, the technical skills as well, to be able to produce that, because you can give the parts to people. And I have in the past, we wanted to see how when you give the plasmid to people, how they can really express it. So we had some people in our lab where we just gave the parts to them to kind of attempt to express it, and it's been quite problematic. So even in the same lab where some have been able to express it, you give that and protocol to other people, and sometimes it doesn't work. So we need to actually have that training where you take them through the whole process of expression, and then when they go back, they should be able to do most of these things. And I think from the beginning, when we started with Reclone, where we're looking at the cellular reagents, one reason why it wasn't being accepted was because of all the debris that comes with the cell when you're running a PCR directly with the cellular reagents. And so I think the purification bit is also important. And so if we have this training where you not just here's a part, here's a protocol, go do it, but then if you kind of have that training with them, I think we think that it's going to go a long way to get people to really use the parts and have the full benefit of what we intend it to be.
Yeah, I mean, I personally agree that having something shown to me and doing it practically is always helps it sink in for me a bit better. Any other further thoughts about how to help others, Aaron? you want to?
Yeah. Yeah, I think for us in the Philippines, the one of the biggest problem is actually the bridge between the research and the actual commercialization part. So that's why actually in our project we're kind of emphasizing on, because you can, producing enzymes is one thing, but producing enzymes in commercial quality is another. So since the industry here for enzymes is not that established, so we would like to like demonstrate to the people here that we can actually produce enzymes that are actually high quality and are sellable and not just, you know, lab, like mini prep, for example. So that's what we are focusing right now, actually. And yeah, that's from research to commercialization. That's the biggest problem. So I guess also maybe reaching out to potential principal investigators or research teams there that would like their science to be commercialized and teach them how to do it. Maybe things like that. I think that's the crucial part, at least in our country.
It's always useful to hear how different regions, different countries have different perspectives of reaching people and making best use of the regions that are being shared. Any thoughts from Diego? Yeah.
Yes, no, in the same line, and the other students in our case, in Argentina, you can find two different kind of laboratories. Laboratories we have access to economic support to buy everything they need, and laboratories that don't have the support. But in both of the cases, they always have limitations to introduce to the country reagent or enzymes, everything that is coming from other countries, to importation of reagent in general. There are so many limitations and so many problems. So in this context, the solutions that Reclone offer could be relevant in this region. However, I think that we need to show and we need to demonstrate a robust correlation between in all the stage of the process, the plasmid production, the protein expression, and the protein, the enzyme activity. Without this ability to show this, that the process is robust, and that once the receptors have the enzyme, they can work with the enzyme, like was the case that Romi showed with PFU. But with all the collection, we need to show clearly that the process work with the robust enough to be a relevant tool in the day-to-day of other labs that are not able to buy this kind of enzymes, because all the questions that we receive when we are showing our work with Reclone is what happened about the expression, what happened about the activity, and at this moment we are not able to demonstrate how easy or not easy is the expression and how efficient or not is the enzyme that we are able to produce. So this is our situation right now.
Definitely all very important things is being able to demonstrate that what the reagents are shared actually do work as designed. I know it's already been quite a long road to try and get the different parts out to each of the hubs. Our plan moving forward, and I think Felipe is on the call as well. So Felipe is working with us at the Open Bioeconomy Lab, and he's the main one behind kind of replicating all the collections we have here so that we can distribute out to the different hubs so that there could be subsequent redistributions from the hubs. So we've been trying to troubleshoot how best to kind of scale up the collections we have so that we can do that redistribution and so on and so forth. So definitely recognize the limitations of personnel, trying to be able to test that the collections are correct and active. But I don't know if Felipe is able to say a few words on this about what our next phases are. But we're hoping by the end of the month then, fingers crossed, to have these collections. So our initial collection, so the research and diagnostics collection, the open enzyme collection, open reporters, and open enzymes collection. We're hoping to send these to the hub so that they have our current available collections there for use and for redistribution. But making sure that so the majority of the collections are sort of parts where you can just plug and play, that you can just have to plug together and assemble into full constructs. There's only the research and diagnostics collection, which is ready to express. So we're hoping to work with collaborators globally to help try and make more of the collections kind of just ready to express. And then all our fellows will have a job on their hands trying to help us clone, transform, and stock all these for redistribution. So we're currently in a pilot phase, but hopefully we can start scaling up what we do in the near future. Any other questions that people want to raise? Joseph, hi.
Hi, thank you for the overdose of inspiring experiences from global speakers. I want to ask something about producing enzymes, but with more sustainability and with some purification techniques. So lately I've been researching about it because in order to produce enzymes from reagents and plasmids seems to be, as you said, in a lab scale feasible, but if you speak about it industrially or scale up is very difficult. So there are some enzymes that are produced like in pineapple stems, proteases that are very well, they are rich in proteases that, and this part of pineapples as an example is very easy to obtain and produce enzymes or extract enzymes from. So what is the possibility that you could look into these kinds of enzyme production and extractions and also use it in a reclone projects, etc.?
So sorry, got distracted by a cat. I think it'll be really interesting to look into different proteases and how they can be used to extract enzyme. I'm not very familiar with that strain at all, but well, about those enzymes at all. It's I think usually with the community is a kind of a problem of personnel, whether we have enough people and time and resources to look into that deeper. But happy to, if you post more information about this on the forum, we're happy to dig into it and see if there are any community members that are interested. Any thoughts from anyone else? Anyone familiar with using these kind of proteases from pineapples, you said?
Yeah, I think well, our industry partner is kind of interested on that. So actually, that's one of the things that we thought about. Like, oh, we need proteases and pineapples are rich in them. However, it's kind of difficult though. The extraction part, it's the logistical difficulty also to upscale it is a lot more difficult than, of course, just extracting proteases from microbes. So we choose the microbes route. So that is my personal experience on proteases.
Are you doing particular research into this, Joseph, then, or is this something that you just come across? I appreciate that comment, Daniel. It's always pretty.
Just a quick comment. It is that E. coli is great, but it's limited, and it would be fantastic to make our network grow with other hosts. I understand that some people are very optimistic about what can be done with Pseudomonas and Putida specifically, and Bacillus subtilis as a gram-positive model. But those are microbes, of course. Plants is too far away from my field to have an opinion, but I'm sure it has some advantages. And if that idea of the pineapples for proteases can be probably extrapolated to other organs of some plants that accumulate some kind of specific proteins, and then you recombine for that specific protein with that regulation, but for another protein, and if it is viable, it will be useful. It's a nice idea, too far from my field to have a scientific opinion on it, but it's food for thought, at least.
Definite food for thought, indeed. I think Adrian, I wasn't sure if that was meant to be to me or to the group, but he also mentioned pina coladas. So I feel like we might need to get a few more plant-based scientists within the community to shed more light on this. Cool. Any more thoughts from the community about enzymes that we could potentially add to the collections and how to best make use of them?
Yeah, I think it would be nice to have some antibodies though. So yeah, it's good for ELISA for quantifying the proteins that people might have produced from our enzyme collections.
I mean, antibodies would be also quite useful to make use of if we can. Adrian, sorry.
Oh, yeah, I was thinking that it would be good to prioritize the enzymes. Sorry, not the, well, yeah, the enzymes that we are needing for assembling, like Moclo specific enzymes there, because then if those would be available, then we can do more assembly at different labs and actually kind of use these ones across the Reclone community there, because that's pretty would be a pretty basic editing feature. Those enzymes are so easy to come by.
I mean, I think Scott's on the call here as well. I think he was in contact with oh god, I forgot her name now. Ellen Jorgensen, who's trying to make some more plug and play using the open enzymes collection into Reddit Express plasmids. Is that correct?
Ellen actually has she got a grant from experiment.com to be able to work on this as a project. So she's putting, I'm not exactly certain what construct she's putting together and what her plans are. I'll also add, since Adrian's interested in enzymes involved in Moclo, I did get a grant of 20,000 base pairs from Twist, well, from iGEMs through my work with Twist. Sorry, today's a tough day. It's our first thing in the morning, so I'm a little bit foggy. I'll say that again. I got a grant of 20,000 base pairs from iGEMs for gene synthesis at Twist. And I did synthesize the BSA1 and its 2 methylases. So I've got 2 of them cloned and we'll be doing whole plasmid sequencing this week or next week. And the number 1 methylase, M1, it's being a pain to clone into the POpenV3 vector. So I'm working on a basic SIVA vector to put it into, and I'll hopefully get that in a few weeks or so. But once those parts are made, then I'm happy to share them, made and sequenced. Happy to share them. So yeah.
That's fantastic to know, because those are very, very important. Yes. Thank you so much. And good luck with the SIVA plasmids there.
Yeah, BSA1 went off patent last year, so I'm comfortable sharing these things. So yeah.
I understand that BSA1, not all the vendors would recommend that for. There are only some vendors that work well for Moclo. Is that correct?
Sorry, I don't quite understand your question.
There are a number of vendors that produce BSA1, but I understand that for Moclo there are some more requirements, and there's only one or two vendors that guarantee that that's going to work.
Well, so there are two enzymes that have the same restriction sites. So they're isoschismers. Thermo Fisher sells one, and that one's actually in the open enzyme collection. The other one is BSA1. Of course, New Biolabs had the patent on that, so they've dominated the market with that, and they've optimized it for Golden Gate assembly. And so I don't know what their proprietary optimizations are, so it's hard to say.
Okay, so you're talking about the Thermo Fisher one.
Thermo Fisher, yeah, those parts are in the open enzyme collection. So it's the restriction enzyme and 2 methylases. And in the new Reclone collection, I did design a few parts that have allow one to make an operon. So it's a synthetic operon. So you can join 2 transcription units, so 2 genes within that. So I'm not certain where things are at with regards to replicating the Reclone stuff. It's when Yankee, do you know anything, or maybe Philippe?
Still work in progress. I think Philippe, he's been trying to replicate the B sub secretion tags that you sent over. But we've been doing the backlog of sending bits and pieces out to people, so I think we will need to get back to replicating those plates again.
Okay.
I think he managed to transform. Yes, I think Felipe did manage to transform some of the collections, but I need to double check on that because it's been a little while.
Okay. Yeah, I think Aaron here has been waiting a long time for the B subtilis in particular. So
yes, I realized that. Yeah, because I think Felipe managed to transform 5 of the B sub plasmids, but the 6th one is proving to be a bit tricky. So he's recently ordered some primers to try and replicate, trying to amplify that from the plates. Hopefully we'll be able to say more on that soon and send all those plates off to Aaron, because I know you've been waiting a little while. So we have just come to time. I want to thank again all our fellows today for giving their talks about what they've been doing at each of the hubs. If people would like to stay on for another hour or so, and it is entirely optional, but we're working on the MIT Solve grant, which is to try and so this is an application that was due tomorrow, but it's been extended to the 23rd. And we've been trying to prepare the application to increase pandemic preparedness and health security. And this focuses on the application of the hubs. And we use explain more use of the research and diagnostics collection and how that can be helpful in diagnosing diseases in low-income regions. So if folks are interested in helping out in this, let me share this in chat.
Yes.
So I posted that in chat, and this is what we'll be working on for the next hour if people want to stay on and help work with that. I know some of you in the group have helped us out of this application so far, so thank you for your contributions. Otherwise, feel free to get on with the rest of your days or have a good evening, I guess. Thank you very much.
Great. Thank you. Have a good day.
Thank
Good
you.
day.
Nice to meet you.
Thanks a lot.
Good day.
Ciao, ciao.
Thank you. Bye-bye.
Bye, everybody. I mean,
Bye.